So Immusoft is developing B cells as biofactories for extended exposure and expression of protein therapeutics. And more specifically, we're working with a subset of B cells known as plasma cells. And I like to think of plasma cells as kind of the dedicated biofactories of the immune system. They produce and secrete thousands of antibodies per second. And it's that protein manufacturing capabilities of the plasma cells that we harvest at Immusoft, that we harness at Immusoft, excuse me. And so the way it works, we extract the patient's cells, insert our therapeutic gene, we coax them into that plasma cell state, and then we re-administer those cells to the patient where they engraft in the bone marrow and for many years secrete our therapeutic protein. And these plasma cells have actually been shown to be very versatile. We started out with an antibody, an anti-HIV antibody, and we've moved from there to expressing enzymes like iduronidase for MPS I and iduronidate sulfatase. We've done structural proteins like procollagen 7, which is actually a huge protein, follistatin, a signaling protein, factor VIII. I could go on, but there's a lot of excitement around the many prospects of the different indications that we can pursue with our platform. And so the initial indication that we're pursuing is MPS I, a lysosomal storage disease. We've got really great preclinical data showing that in numerous different studies we've been able to reduce glycosaminoglycans in these animal models and increase the expression of plasma levels, in particular, of our enzyme iduronidase. And so we're working real time to get our IND filed and looking to do so in April of this year. So 2019 is going to be really an exciting time for us. And, I've got to give a nod to the team that I'm working with and really proud to be working with. They've done a fantastic job in getting this science to where it is. And so really, really great group. So some of the things that, really compelled me about this approach is when we compare it to some of the other gene delivery modalities, I saw what I felt were some pretty clear advantages. And so we tend to think of, virus delivered and gene-modified stem cell delivered approaches as being one and done. And, I would go so far as to say is, whether that means it's a lifetime of benefit is to be determined, but given present technologies, redelivering these approaches is really not feasible without substantial risk or morbidity. And so in our case, it's an autologous approach. It's virus-free. We can dose our patients, monitor, and then over time, if need be, we can re-administer our therapeutic. And so the way it works, we start out, we apheresis a patient. Ex vivo then, we put our gene of interest into our B cells. We extract the B cells, we put that gene of interest in. We use a Sleeping Beauty transposon system. We then expand and differentiate the B cells into plasma cells. And then we administer those into the patient. And so from vein to vein, the entire thing is about 10 days. There's 7 days in culture, so pretty substantial advantage as well over current CAR-T approaches in particular, which are a couple 3 weeks from vein to vein. And so in terms of our financing, we closed on $23 million earlier this year. That gives us runway through mid-2020 timeframe, enough to dose many patients in our initial clinical study, which is MPS I, and advance our follow-on program into an IND-enabling, IND-ready state. And so with that, we expect to be in the clinic here later in Q2 and planning for success. We should see biological activity then not too long thereafter. And so that's defined as either an increase in the concentrations of our iduronidase enzyme or a decrease in our substrate, which are glycosaminoglycans. And so with that data, planning for success, we'll be looking at going into a Series C and getting that launched in the Q3, Q4 timeframe of this year. And so would be happy to speak with anyone offline if there is interest in learning more. And with that, I'll hand it over to Tom. So do I just join you here?
Sure. Thank you, Sean. So maybe we can take a step back. I think most people in the room know much more about T cells than B cells. So maybe give us a little bit of a sense of, it sounds like your ex vivo amplification is much simpler. And maybe you can talk a little bit about the persistence of the cells, some of the attractive properties of B cells, because I think they're quite different than T cells, and people don't maybe appreciate that yet.
Yeah, well, thank you. So in terms of manufacturing, I mentioned our time frame being an advantage, and so 7 days in culture means that we've got simply faster turn, which is bodes well in terms of scalability. And then once we inject them into the patients, what we do know about plasma cells is that long-lived plasma cells have a half-life of about 17 years. And so we can take a blood sample from a patient who's in their 80s, and we can find antibodies that have been produced to a smallpox vaccine from childhood. And so those antibodies are being produced from plasma cells, which have remained engrafted over many years. And so that's actually key to our approach, is that we anticipate our B cells, our plasma cells, will engraft and last many years, producing therapeutic proteins.
Okay, and you'll probably hate this question, but
fire away.
Everybody's thinking in T cells about how to go allogeneic. Is what you would want to do to a B cell to make it allogeneic? Is there the same sort of thought? Is it thought to be easier, harder, or is it early days?
So what I will say is we've got some work going on to go allogeneic. I'll confess I'm not a scientist, so I can't speak to whether it's easier or more difficult than T cells. But there are applications where allogeneic would certainly make sense. In the initial indications we're pursuing, they are orphan indications, and between that and the fact that our process is more streamlined than the T cell approaches, and again the fact that the infrastructure is constantly improving and becoming more streamlined, we think certainly autologous for the near term works very well for us.
Okay, well, maybe to answer or to think about my own question, with a 7-day incubation and a non-viral construction, is it even attractive to get out of autologous, or is autologous the way to go?
So, great question. Thank you. I think when we start contemplating larger, potential blockbuster indications with huge patient populations, that's where contemplating allogeneic starts to make more sense. And I would, I guess, just add that we've got a number of different directions that we can take this approach, given all the different protein types that we've already shown we can deliver. So at some point, absolutely, it makes sense to start thinking allogeneic.
And then another question on the plasmid side. Can you talk at all about what that does to your potential costs? Because in any cell therapy with a viral vector, huge costs associated. Are they very different for a plasmid construction route? Very
different. Yeah, thank you. That's a good point. So plasmid, in comparison to virus, is a fraction of the cost, which is certainly not unimportant. I would add, though, just from a CMC complexity standpoint, plasmid manufacture is dramatically less complex than virus vectors. And so, my previous life was in the virus world, and when I came over here and looked at the delta between the two, it was a sigh of relief that we are able to work with the plasmid approach, and it frankly works better in B cells than do viruses anyway.
And we're maybe a little ahead of ourselves. Maybe talk a little bit about manufacturing, how you plan to proceed once you're commercial.
Yeah, great question. It's a little ways off for us, but at present we're working with a contract manufacturer, so that gives us great flexibility at this stage of the company. As we proceed, I think we've got options to potentially partner with groups that have manufacturing capabilities, and by that specifically, I'm thinking more about the cell processing side than the plasmid production side. So yeah, options to partner with groups that have their own abilities, and as we contemplate the prospects of potentially raising a lot of money, building out our own infrastructure, or maintaining a relationship with a contract manufacturer. It's a little too early for us to make those decisions, but it's not too early to start kind of wargaming that out.
Okay. So one of the tragedies of a lot of enzyme replacement therapies is the ramifications in the brain. I know you have some thoughts on that. Maybe tell us how you'd attack that problem.
Yeah. So indeed, in the case of many of the lysosomal storage diseases, they do have CNS manifestations that we hope to address eventually. So our initial approach, we're not anticipating great penetrance across the blood-brain barrier. We are nonetheless working on strategies to do that, and in fact, we've received a grant, which I'm not normally too excited about, but it's a $3.5 million grant from the NIH that is largely focused on crossing the blood-brain barrier. So we're actively working on that, and I think that actually will open up many additional opportunities for us.
So another science question to you. But T cells were thought not to cross, but now are known to cross. What about B cells? Do they get in at all, or?
So it depends on the indication, and we know that in multiple sclerosis in particular, T cells do cross the blood-brain barrier. So that's interesting. There is a reservoir of CD19 positive cells in the brain as well that we certainly find interesting. So that having been said, we don't have evidence yet that says when we inject systemically we get our B cells across the blood-brain barrier, but we've got strategies for pursuing that.
Okay, and then a couple of sort of safety questions. The thought about immunogenicity, a lot of these people will be lacking these enzymes. You start to put cells in. Just your thoughts. Is the FDA concerned? Are there extra tests? Just how is that going to play out?
Yeah, so we actually didn't get any feedback on that particular topic from the agency. So one of the things that we know about our initial indication is the approach with the enzyme replacement therapy. There are sometimes immune responses to the enzyme, but that's given in a 4-hour, I guess, infusion. I like to term it as a bolus, but it's an infusion over 4 hours, and so the enzyme levels spike and dissipate. In our case, what we're anticipating is a much lower level of the enzyme, but being produced 24/7, 365 days a year. So conceptually, that makes it less immunogenic. And then because we're delivering from B cells are believed to be tolerogenic as well, given that every B cell essentially is delivering a non-self protein, a unique antibody, and so there's some expectation that the B cells will be tolerogenic. And then on the slide, the advantages over some of the other approaches. There was mention of us delivering a defined cell population, a defined cell type. And so what that means is that there's actually a product on the market, Velcade, that selectively gets rid of plasma cells. So we don't think we'll need it, but there's an opportunity to reverse this if there were a need.
Okay, and then just one final question. So you have memory B cells. What happens in an infection if one of your B cell clones takes off? Is that a concern?
Yeah, great question. Thank you. So we've got, we're delivering very highly clonal populations of B cells and plasma cells back into the patients. And so if there were a chance that an antigen cross-reacted with one of those B cells, it's less than 1% of our B cells that could potentially ramp up and expand. So there's no real concern about the potential for a massive expansion of our product.
Perfect. Okay. I'm sure Sean would love to answer more questions offline, but thank you for coming in.
All right. Well,
Great
thank you,
story.
Thomas. Appreciate it.